Okay, well, I'd like to thank the organizers for having me here today. When I was putting this talk together, I looked back at the title of my abstract, and I realized that not only could I have spent more time maybe making it a little bit more exciting, but it only actually talks about it only describes half of what I'm going to tell you today. So what I'm trying to get at in the title is that we've actually developed this novel assay that couples RNA in situ hybridization with flow cytometry in order to detect cells that have been co-infected with two different strains of hepatitis C at the same time. Now, the second part of my talk is why we're actually interested in finding those co-infected cells, and that's an idea that's been developed in the Kierkegaard lab called the dominant drug target. Now, in order to select for drug-resistant viruses, you need two things. You need mutation and you need selection. And we know that HCV mutates rapidly through low-fidelity replication with the NS5B polymerase. And we know that during certain antiviral therapies, you can select for drug-resistant variants out of this quasi-species, which I've depicted with this rainbow of new genomes. And if I had a pointer, I could show that to you. Okay. And so we think about all these different mutations in this co-infected cell as different alleles. So some of the mutations are going to be dominant, some of them are going to be recessive, just like other alleles. What I want you to think about is in the context where we're selecting for drug resistance, we think that this is a situation in which drug resistance is the dominant allele, and all the drug susceptible mutations are then recessive. We can then select for drug resistance. And the question that Carla had was, why do we always think that drug resistance is dominant? Are there any situations in which drug susceptibility could be the dominant strain in these mixed infections? And what would happen then? If you add a drug to something where the drug susceptible virus is dominant, you could then potentially repress the outgrowth of all viruses, including the drug resistant ones. And this is what we call a dominant drug target paradigm. And it's a situation in which we think we could choose viral targets that induce dominance of drug susceptibility. And we want to do that through what we call, we think that one of the hallmarks of a dominant drug target is something that's highly oligomeric. So if you look on the right, you'll see potentially the capsid of the virus. So not only do the genomes mix in these mixed infected cells, but all of their proteins mix as well. And so in this situation, our red antiviral genome, drug-resistant genome is still producing proteins, but they have to arise in the context of this mixed oligomeric structure where you still have drug-susceptible capsid proteins in that mixed oligomer. Now that mixed oligomer could potentially inherit drug susceptibility, still bind antiviral compounds, and you could repress the outgrowth of these drug-resistant strains in the context of these mixed infections. And our preliminary evidence with poliovirus, this is research done by one of the graduate students in our lab, Elizabeth Tanner. On the left is a rendering of the poliovirus capsid, which I think really captures the idea of the dominant drug target paradigm. So you can see that in red, we've got drug-resistant capsid subunits, and in gray, we've got drug-susceptible capsid subunits. And the little brown squiggles are our antiviral compound called VO73, which targets the poliovirus capsid. I think that you can really see that even though drug-resistant capsid proteins are in this complex, there are still drug-susceptible subunits there, which are binding drug, and the mixed capsid as an entire complex is inheriting drug susceptibility. And our data support this idea. So in the first column on the left, we've got a strain of poliovirus that's resistant to VO73. When you grow it in HeLa cells on its own in the presence of VO73, it grows quite well. But as you titer in escalating doses of drug-sensitive virus, you repress the outgrowth of all viruses, including drug-resistant ones in those cells. So at this point in time, the dominant drug target idea was kind of gaining some steam, and Carla wanted to expand this idea to other viruses. So we've got another postdoc in our lab that's working on dengue, and I chose to expand this idea to hepatitis C virus. But one of the key components of actually studying dominant drug targets is having co-infected cells that are infected with a drug-susceptible and a drug-resistant virus at the same time, so we can mimic that situation in which the drug-resistant virus first arises. And so Elizabeth was able to do this by using high MOIs of 100 or MOIs of 10. And for any of you with experience with the JFH1 strain, you'll know that an MOI of 0.1 is probably more likely, or even in some cases an MOI of 0.01. So the problem becomes, if we use an MOI of 0.1 as an example, how do we find co-infected cells? First of all, you're going to have a lot of uninfected cells. Maybe 1 in 10 cells will be infected with your drug susceptible, 1 in 10 will be infected with your drug resistant, and if you do the math, maybe 1 in 100 will be co-infected, and it's probably worse than that. And so we decided that we were going to utilize an RNA in situ hybridization technique in order to differentiate between these two viruses at the RNA genome level. This technology couples target-specific probes which bind your RNA of interest and use a branch DNA technology to amplify your signal, and it puts about 8,000 fluorescent molecules onto each one of your target RNAs. So when I called Affymetrix and I told them, okay, I want probes that differentiate between a drug-resistant and a drug-susceptible strain, they just laughed at me and said, well, the probes aren't specific enough to differentiate between point mutations. So I was originally a bit deflated, and then I fell back on some of my work in graduate school, and I thought, okay, well, what if I alter the codon structure or the RNA structure of JFH1? And then I can have two different strains that are differ through mutation. And so I chose 3 different regions of the JFH1 genome in order to induce these codon alterations. Now, again, we're mutating the wobble base pairing in these codons, so we're altering the RNA structure, but we're not altering the protein structure. And so I'm going to focus on this mutation that's centered on, that's in the C-terminal half of NS3. We were able to incorporate 247 mutations over a 918 nucleotide stretch, and then Affymetrix was able to design probes that differentiated between these 2 strains. And so when we got these probes, we first did confocal microscopy, and we can actually differentiate these strains based on positive strand probes and negative strand probes. And so you'll see type 1 is our wild type probe set, recognizes our wild type virus, but not our codon-altered virus. And type 6 is our codon-altered probe set, recognizes the codon-altered virus, but not the wild type virus. The next part of this, I got some help from a postdoc in Gary Nolan's lab at Stanford, and we took this RNA in situ hybridization technology and applied it to flow cytometry. So these are HUH 7.5.1 cells that are transfected with either our wild type virus, our codon-altered virus. The third sample is one where I've mixed the 2 singly transfected cells into the same sample and run them through the cytometer. And in the 4th sample, we've got co-transfected cells. So you can see that when we're measuring with our wild type probes on the y-axis and our codon-altered probes on the x-axis, you can see that our wild type probes, again, recognize the wild type virus. The codon-altered probes recognize the codon-altered virus. And in our mixed, we were able to define where the 2 single positive populations were on the same plot. And we were really excited when we saw that our co-transfected all localized into a separate population that we could resolve. And so now we've got this mechanism of defining which cells are co-infected and differentiating them from all the singly infected cells. And we want to use this technology to quantify dominance and identify dominant drug targets. So if we go back to the dominant drug target idea, what are the different targets that you have during an HCV infection? Obviously, the capsid was a really great dominant drug target in poliovirus. So we're really interested in anything that targets the core or maybe the E1 or E2 proteins. PAV617 is a compound I've been playing with, but I'm not going to tell you anything about that today. The protease is something, obviously, the protease inhibitors have gotten a lot of attention. We think that these are your stereotypical non-dominant drug targets, and this is kind of supported in the literature where patients are getting drug resistance to these. The NS3-4A functions as a single subunit. It's not part of a larger oligomeric complex where it's going to be exposed to dominant negative effects by other proteins within that complex. The other 2 targets that we're really interested in are NS5A and NS5B. I don't have time to talk about the polymerase today. We are interested in these nucleotide analogs. We think that depending on how the inhibitors function at the polymerase level, based on our work with poliovirus, some of these inhibitors could induce dominance of drug susceptibility. But again, I'm not going to talk about that today. I'm going to focus the rest of my talk on an NS5A inhibitor that we received from Tim Tellinghausen at Scripps. It's called SR2486. Again, the reason that we're interested in NS5A, it's highly oligomeric. There's a paper from 2004 from an Italian group that shows that a non-functional NS5A can actually have a dominant negative effect in mixed replicon experiments. And also these inhibitors function at really low concentrations. So some of them function at picomolar concentrations, and if you think about that, there's actually less compound than proteins, which really kind of fits the paradigm of something that functions in trans as well. Okay, so I've just got 2 slides left. This is our NS5A mutant. It's called Y93N. This has been characterized in the literature. It's in domain 1 of NS5A, and it confers resistance in the presence. We're using 500 nanomolar SR2486 in our experiments. This is my final piece of data. So we wanted to use flow cytometry and our flow RNA assay to really kind of see if we could measure dominance in a mixed infection. So we're using our codon-altered virus as our drug-susceptible strain. We're using the Y93N mutant was cloned on the wild type background, so we could differentiate between these 2 viruses using flow RNA. Again, we've used a mixed sample in order to define where the single positive populations came up on our plots. And then we looked at co-infection. So if you look in the first column first, in the presence of DMSO, we were able to infect about 5% of our cells with our codon-altered virus. That population went away when we added drug, showing that this is truly susceptible to compound. And through the help of Lauren Terrell's group, we were actually able to get higher, tighter stocks of our Y93N through culturing in human serum. So we're real excited about doing future experiments where we can actually get higher levels of infection. But we're able to infect 54% of our cells with our drug-resistant strain. And when you when you culture those in the presence of 500 nanomolar SR2-486, that drug-resistant population persists in the presence of compound, as you would expect. So we used our mixed samples to define where we thought the double positives would show up on our plots. We were really excited to see that 2% of our cells were co-infected. Were co-infected in this experiment. And when you add drug, we lost 2 of the 4 populations disappeared. So the singly infected drug resistance still persisted. The singly infected drug susceptibles went away, but we were really excited to see that the double positive, the ones that were co-infected, also disappeared. So this is preliminary data. There's obviously a few different interpretations to this, but one of them is that NS5A drug susceptible proteins that are drug bound are having a dominant negative effect on drug resistant NS5As in the same cell. Obviously, this is preliminary and we need to do more work on it, but we're pretty excited about NS5A as a potential dominant drug target in HCV. So with that, obviously we want to quantify dominance using flow RNA. We can do this at the population and at the single cell level, so we can use flow cytometry to isolate our co-infected cells and do things like nested PCRs to quantify how much of each genome we have in each cell. I think that also using this human serum technique from the Lauren Terrell's group is going to help us get higher levels of co-infection. Once we found a dominant drug target, we're really interested in mechanisms of dominance. And the big question is can this dominant drug target idea translate in vivo? Our experience with poliovirus so far, the VO73 capsid inhibitor actually does repress the selection of drug resistance in the PVR poliovirus receptor mouse model. But again, different viruses, different life cycles. And then again, we're always looking for new anti-HCV compounds, things that target highly oligomeric structures that would fit the dominant drug target model, so things that target the core or the E1 or E2. With that, I just want to thank my lab, specifically Carla, my mentor, Elizabeth Tanner, who did the poliovirus work, the Terrell lab, Lauren himself, and then Mike and Reneke, who helped me with the human serum. Yuri Goltsev is a postdoc in Gary Nolan's lab, who got the flow RNA technology kind of off the ground. And then everyone else, because I'm the only person in my lab that works on hepatitis C, so I'm a member of the NCRTP. This whole community, as well as my mentor, Dr. Chris Richardson, have been really helpful in giving me hepatitis C specific information, as well as Selena, our previous speaker, used to be right next door to me and knows lots about hepatitis C, and so I miss having her next door. But okay, with that, I'll take questions.
Thank you very much. We have time for one question.
I have 2.
Okay, 2.
First of all, not all codons are created equally, right?
Yes.
So how can you, I think you have to be careful there not to introduce a confounding factor.
Yeah, so I created
levels.
3 different codon-altered strains. Yeah, I mean, you're definitely worried about hitting things like secondary RNA structures that are...
No, I'm talking about availability of tRNA. So there are different levels of different tRNAs.
Yes.
So you may have introduced a fitness difference.
So I didn't show any of the fitness data. This of the 3 strains, I had one that didn't grow at all, and I had one that kind of had an intermediate growth phenotype. The mutant that I've chosen, the growth curves and everything fit the same as our wild-type JFH1, at least in culture. Okay. Rello Andino's group at UCSF did similar experiments with poliovirus, and they definitely saw that the codon-altered viruses had different growth characteristics in vivo, but they were the same in culture.
If I understand you correctly, you've made your mutants in the absence of the drug resistance mutant and compared them?
If you compare my codon-altered strain that I showed in that experiment with wild-type JFH1, they grow at the same level in culture anyway, and you can create continuous cultures that last months, just like you can with wild-type.
So then my very quick second question is, and it's not a question, it's really, you're working with wild-type JFH.
Yeah.
So it's going to continue to mutate, I promise you. I've done this 50 times.
Yeah.
So at some point you may lose your detection of one of your strains, because you will get adaptive mutations.
I always create my stocks through transfected HUA 7.5.1, so they're pretty close to. I don't actually continuously reinfect and create cultures that way. I always start with a transfecting.
But how long do you grow them for?
Maybe with human serum, I've been going a little bit longer, 2 months. I mean, yeah, I understand.
You could have
I understand there's
mutations.
I understand that there's mutations.
Yeah, so you'll definitely it will mutate.
Okay.
Like after 2 days you'll have mutations.
Okay.
I promise you.
Okay. Thank you, Nicholas.
Okay, thanks.
We have to go on.